DNA, given by Miss Davis. So the purpose of DNA is it is the directions for life, all life. It is for the directions, more specifically, for how to make proteins. And if you recall, proteins are how life happens. For example, proteins are for growth and repair. So if you get cut or you scrape your knee or you break your arm or your leg, you need to repair. And also when you're a little one, you grow. And so proteins make that happen. It's also for communication and feedback. So communication meaning I'm hungry, I'm tired, I need to walk. Or feedback, I'm touching a hot stove and I need to stop doing that. Or I need to become tired so I can fall asleep, and I need to make certain hormones, which are proteins, in my body so that I can become tired and fall asleep. Or I'm hungry, so I need to have certain hormones to tell my body to start digestion and saliva production and find food. So those are some of the basic things that we do to live, and that is all a product of protein. And then finally, maintenance. So, maintenance meaning your cells just get old and you need new ones. It's not that they're ruined, it's not that they're broken, it's not that you've cut your knee or broken your leg or you have some terrible disease. It's just simply living, they wear out, you need new ones. And proteins help all of that happen. And if you look back, proteins, the direction, are in DNA. All right, so first we need to look at the structure of DNA in order to understand how this slide, the directions for life and making proteins, you got to look at the structure first. So there are really three main parts. The sugar, which is a deoxyribose sugar. So you need to be able to say that, deoxyribose. There is a phosphate group, and there is a nitrogen base. Now, there are four types of nitrogen bases: guanine, cytosine, thymine, and adenine. Those are the four types of nitrogen bases. The three of these things, A, B, and C, sugar, phosphate, and nitrogen base, together make what we call, let me write this down. This is called a nucleotide. A nucleotide. So when you're referring to a nucleotide, what you're referring to is the sugar and the phosphate with some base attached to it, one of those four, a nucleotide. So please write that down, and maybe you can make this little thing saying that these three things make up a nucleotide. Now, there are other parts of a nucleotide, and other functions of the structure of the DNA. So something that we talk about with the sugar and the phosphate, we call this a sugar-phosphate backbone. And so when you're looking at the structure, you've got this long strand with one of these four bases in between. I'm going to come back to this slide in a moment, but I want to look at this sketch because I want you to start sketching this, and so I'll leave this up here for a while. You can also pause this if I go too fast and you need to actually stop and sketch. But if you look at this blue ribbon, see this S? That's S for sugar, that's P for phosphate, S for sugar, P for phosphate. So it goes back and forth, sugar, phosphate, sugar, phosphate, sugar, phosphate. Remember, when I say sugar, specifically, I'm talking about deoxyribose, the name deoxyribose phosphate, deoxyribose phosphate. And if you notice, this is kind of the outer edge of both of these strands, and that's why we call it a sugar-phosphate backbone. Right here, guanine and cytosine always bond together, and thymine and adenine always bond together. From a chemical point of view, the type of bond is called a hydrogen bond. So when you're looking right here, see how A and T, that's adenine, and this is thymine, and there's cytosine and guanine. They always bond together. So if you look on this diagram, purple is always with pink, and yellow is always with green. And when they bond together, they make hydrogen bonds. Now notice this word, it's telling you that it's weak. It's a weak bond, and we're going to talk about why a weak bond might be valuable later. But I do want to point something out. One of these bonds is weak, but lots of these bonds across here end up becoming strong. So it's almost like if we think about one small thread, you could break quite easily, but when you weave it all together to make a fabric, that fabric is pretty strong. That's the same concept with DNA and the hydrogen bonds. So I want to talk about an analogy with phone numbers and DNA, and this it will give you an idea of how huge DNA actually is. So phone numbers. How many options are there available? How many options are there available? I'm going to go out of this for a moment and fill it out this way. There are 0 through 9, right? If you think about your phone number, 916-424-1342, right? There's an option of anywhere from 0 to 9. So we've got 10 options, right? 0, 1, 2, 3, 4, 5, 6, 7, 8, 9. Now, how many slots are there? Look, you have 1, 2, oops, something like this here, 916. I'll make up a number here. I don't know, 4, 5, 3, 2, 3, 2, 4. How's that? If that were somebody's number, if you look here, there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. There's 10 slots. Okay, I'm going to go ahead and delete this because that's not really what I wanted to show you. There are 10 slots. Now there's a mathematical equation. If you take the number of slots available and take that to the power of 10, let's find a calculator here. Here we go. And we clear this out. So if I take 10 and I raise it to the power 10, I get, what is this? 1 million, 10 billion options. I'm going to actually write this out, 10 billion. Or rather, what is that? 1, 0, comma, 1, 2, 3, comma, 1, 2, 3. There we go. 10 billion possibilities for phone numbers. Do you think that that is a sufficient number of options for all United States people to have a phone? Because think about it. We have a house phone, many of us have cell phones. We might have a work phone. And it's no worry because we've got 10 billion options out there. Now let's talk about DNA, because DNA is a lot like a phone number. How many available options are there? Now, when I'm talking about options, what I'm talking about, if you look at this slide, these are your options. And you've got 4 different base pairs. So we're going to say that you have 4 different options. How many slots? So how long is this DNA? How many slots? Like I have T, there's 1, A, there's 1, G, C. How many slots does the human DNA have? Well, guess what? It is 3.3 billion. 3.3 billion. That's a huge number compared to 10. And so if we do this mathematical equation, we take 4 and we raise it to 3 billion, and we do this on the calculator, watch what happens. So we say 4 raised to 3 billion. So it's 1, 2, 3. 1, 2, 3. That is how many slots there are. Guess what we get? Infinity. Is it actually infinity? No, it is an actual number, but it's so huge that who cares? So what we're going to say is the answer to this is infinity. And what that means, oops, I can't spell, infinity. What this means is that because DNA has so many slots, and even though there's only 4 different ways, 4 different things to put in a certain order, the number of possibilities is infinite, meaning that all living things are not going to be completely identical because there's infinite possibilities. This is why maybe humans look similar, but we are actually different because it's just statistically almost impossible to be identical. I find it quite amazing that there are only 4 different chemicals put in a different order that give us the human. You realize that these same 4 chemicals put in a different order, maybe with, I don't know, 2 billion, you might get a piece of grass, or you might get a flower, or you might get a zebra, or a mouse, or an insect, or a whale. So, I mean, I just named off all these random living things, and it's pretty spectacular to think about the fact that just an arrangement of 4 different chemicals will give you such a diverse amount of different living things. All right. So, the analogy for a recipe book in DNA, I'm going to give you another analogy. So, a recipe book has lots of recipes, but you really only need a few at a time, right? You don't need the whole book. And another thing you can do, you can photocopy the recipes and you can share, right? You can give some to your mom, your sisters, your uncles, your aunties, your brothers. Or so you don't destroy your book, right? So if you're in the kitchen and you've got a recipe book, a lot of times what my mom does is she'll photocopy the recipe, then put that photocopy on the counter. That way when you spill your tomato sauce all over it, you don't ruin it. So this is a lot like what DNA does. DNA has a lot of recipes for how to make proteins. Remember the importance of proteins. It's how we are alive. And oftentimes your body only needs a few at a time. So you can make a copy, and in our body we call that copy RNA, and you can give it to other parts of the cell to make the protein. What's interesting is that the DNA never leaves the nucleus because it's so precious. We don't want to mess it up. Just like that recipe book, it's kind of precious. You don't want to mess it up by pouring stuff accidentally all over it. DNA is the same way. It's protected inside the nucleus. So the way that the information comes out is by making a copy. And then the cells can do with it what they want and make the proteins. So how does DNA hold information? It holds it by the order of the nitrogen bases. So remember how I was talking about there's 4 nitrogen bases and there's 3.3 billion slots for a human, but maybe you put it in a different order, you might get a slightly different human, or you have maybe less slots and you might get a different type of animal. It is by that order, G, C, T, A, or A, A, T, G, G, C, C, A. You know, you can put them in various orders and you get a different creature. This is a lot like a phone number, right? So if you put the numbers in a different order, you're calling Miss Davis, and if you change that order, maybe you're calling Mr. Chapman, or maybe you're calling your mom or your auntie. So it's kind of like that. Different order numbers give you a different person. So a different order of nitrogen bases gives you a different creature. So how is it accessed? It is accessed by unzipping a section, making a copy that is then sent out of the nucleus to the rest of the cell so it can make that protein or make proteins. So only a small section of the DNA needs to be unwound. Let's go back and look at this slide here. In fact, I want to show you something. What I mean unwind is, remember how we talked about this weak hydrogen bond? Well, you can come in here with a special chemical called an enzyme. It's a zipper. It's like chemical scissors, and it can unzip a section of this DNA. So what it will do, it's like it chops it right here, and it can unwind. Let me get a better graphic for you. If I go here, unzip DNA, and let's look at the images that come up. Sorry, it's taking a long time. Here we go. Here's this section. This one's a good picture. So what happens, see this blue, this is the original strand, and something came along and unwound it. So if you look, see, here's the original blue, like that. And that makes it so that you can make a new copy of it. and that can then be sent out. You get the idea of unzipping. And so when you look at this picture where it's going to be cut, this specific order is really important. Maybe I only want to access this small section because it's going to make the hormone that tells me I'm hungry. Well, you don't need the whole darn DNA, you just need a small section. And so when you're looking at um, how is information accessed, it's by accessing a specific section. Let's relate that back to a cookbook. If I only want the recipe for how to make lasagna, but I don't really care about how to make um, a Greek salad, I only want to access the one recipe, right? That's just like our body. So it only accesses the section that it needs by unzipping that small section, by breaking those weak hydrogen bonds, making a copy, and then sending that copy out to the body. So there's a lot more we can talk about with DNA, but this is a nice intro, and I hope you enjoyed my presentation. Thanks.